So what I'm going to do is to talk about clinical trials. So hopefully this is not advancing. So let me see if I can. Okay. So the purpose of clinical trials is to allow us to address the outcomes of intervention under experimental conditions. So Dr. Kim started off by saying that ideally we want to be able to do clinical trials because that's the cleanest way. On the other hand, it is expensive and it takes time. So the proper way of addressing whether an intervention leads to a certain outcome is to do a clinical trial. You can have one arm, you can have multiple arms. In general, we assess whether one is better than the other, but occasionally you also do non-inferiority studies. It can be open label, it can be blinded, and it can have multiple designs. But I won't get into the new novel study designs like adaptive designs or smart trials because those are a lot more complicated, and you need to have very good statistical support. So in industry, for pharmaceutical studies, generally speaking, we hear about phase 1, 2, 3, 4. So phase 0 is before it gets to humans. Phase 1 is very early stage, and here oftentimes you don't even include patients with the specific disease. It's oftentimes healthy volunteers. You just want to test different doses to look for safety. And well, this keeps moving, okay. So it's mostly safety and pharmacokinetics. And that allows you to go from a very wide range in doses to a smaller range, and you get into phase 2. By the time you get to phase 2, you're trying to figure out what's the optimal dose, what's the optimal duration. You certainly want to confirm safety and also get a sense of efficacy. The phase 3 is really when the industry go to the FDA or the European equivalent to get approval. Here, you have already narrowed down to what is the dose that you think will be safe and effective, but what you want to do is really enroll a much larger number of patients, a little bit more diverse, to prove that a drug is safe in a more broad patient population, that it is effective, and you might do subgroup analysis to show that it works for men and women, young and older patients. Because here is really when you get the proof of the pudding before the drug gets into the real world. We all know that in that setting, you're selecting for patients who meet very stringent criteria. So ultimately, you want to know when a drug gets approved and being used by patients in your clinic, is it still safe? Is it still effective? That's the phase 4 studies. Oftentimes, phase 4 studies will also look for specific patient population that have certain comorbidities or certain characteristics that you don't want to include in a phase 3 study. So you need to prove, for example, with Hep C drugs. Early on, the HIV patients were separately looked at. The patients with CKD were separately looked at. So your phase 4 studies need to include those patients before you can conclude that they continue to work. So I hope that this, okay. So when you're trying to design a clinical trial, there are several key questions. The first question is, well, what is the question you try to address? What do I want to prove? What is my hypothesis? What are the aims? And what are the endpoints? Now, endpoints are the most important element of a clinical trial. You have to be able to specify what, when, and how. And I'll go to show you some examples later on. Then you say, okay, now that I know what question I want to study, how am I going to address that question? Well, what's the trial design? What are the patients that I want to include? Well, I hope that will come back. Okay, what is the intervention, and what's the comparator, and what data am I going to collect? You've designed a study, you've collected the data. Now you have a lot of numbers in front of you. You have to then say, okay, what am I going to do with those numbers? What is the statistical method that I will apply? So you have to think of all these things before you can launch the study. All right. Before we get to pharmaceutical studies, which many of you are not going to be designing because that is very expensive. That takes a long time. Well, my residents and fellows and my junior faculty now get into the cheap studies, which is behavioral study, and it's very popular, okay? We want to change people's diet, we want to change people's behavior, make them move, make them do something. That doesn't cost us much, okay? But one of the things that it is extremely important to bear in mind is it is very difficult to standardize behavioral intervention, okay? And even when you're doing the pharmaceutical studies, we now have to collect the patient's quality of life, patient-reported outcomes, because maybe it's going to really make your liver fibrosis go away. But if I'm going to make you so miserable because you're going to have diarrhea, 20 bowel movements every day, who cares? Because there's no way that in the real world anyone would take that drug. Okay, so with behavioral studies, one of the things that we have to pay attention to is what's the end point? Because you want to change someone's behavior, but how do you define that? How do you measure that? That's very important, but the intervention is even more important. Because if you're doing a pharmaceutical trial and you actually have pills that you've asked the patients to take, well, everyone is taking the same pill, the same type of pill. So it's very standardized. Your intervention is very standardized. But behavioral intervention, just think about it. If your behavioral intervention is nutritional counseling, you want people, you're doing a NAFO study, you're going to give them nutritional counseling, you hope that this is going to change the diet, you're going to motivate them to move more. Okay, so you have a script in which you're going to tell the patients X, Y, and Z. But if you have 10 sites, you're not sure that everyone say exactly the same thing. Your tone, your attitude might actually be a little different. And even if it works, when it gets to the community, it's very difficult to standardize. So behavioral trial, you cannot say that this particular approach works until you've shown that in multiple sites you're able to replicate the same intervention, there's fidelity, and that you see the same outcomes. So that requires a lot more thought in planning. Okay, now we get to randomized clinical trials. That's the gold standard. And the reason for randomized controlled trials is that you want to balance between the intervention group and a control group. Not all NAFL patients are alike. Some of them have diabetes, some of them don't. Some of them have cirrhosis, some of them don't. And if you have more patients with cirrhosis in one group than the other group, it's going to mess up your results. And the only way to be sure is if you're able to randomize them to reduce the bias. But there are problems with randomized trials because in general it requires a large sample size and therefore it is expensive. It takes time. And sometimes if you're trying to compare one intervention to another, you have to be sure that both in terms of likelihood of benefit and risk, there's equipoise. So for example, with Hep C DAA therapy, it was impossible to do randomized trial once we realized how effective those DAAs are. You cannot tell the patients, you'll be randomized to get this drug that is going to give you 98% chance of SVR versus a placebo pill, which is 0% chance. Who on earth would participate in that trial? Well, actually, some companies were very smart, and what they did was they did what they call a deferred treatment design. So the patients are still randomized to treatment versus control. But the placebo group, by the time they get to week 8 or week 12, will automatically receive open-label therapy. That's not so bad, because if you tell the patients that treatment might have a 98% chance of a response, you're only going to just start treatment 8 weeks later, 12 weeks later. That's not so bad because the drugs are very expensive, and the patients know that, well, otherwise they may wait 5 years or 10 years to get those drugs. So that's okay. But equipoise is important. All right. Now, that's the design of a proper study. Many of you are young investigators. You're not quite ready for that. So you can start by doing some feasibility trials, pilot trials, show that it works before you do randomized controlled trials. And even if you have a lot of money, you still want to make sure that what you're thinking is going to work. So what are feasibility studies? Well, the first thing is, trials fail if you cannot enroll patients. You want to prove that you're going to have patients are going to agree to be in a study. You want to show that you can retain the patients. Whatever intervention you're going to introduce, the patients would actually take it and stay through the end of the study. Well, sometimes you're very ambitious. Like Jess said, well, it may be a question very important. You would regret it if you didn't include it, okay? Here's 30 PIs. Each one of them come up with 5 questions. Now, all of a sudden, you have 150 questions. The patients are going to freak out. No one wants to answer 150 questions. So you got to really think through what data is essential and would your patients be willing to go through 100 tests and answer 150 questions? And you have to prioritize them. And sometimes you have to test it out to figure out what the patients will be willing to do and what the patients won't. Well, what about pilot trials? Oftentimes, feasibility trials and pilot trials are being mixed up. They are a little different, okay? Feasibility trials, if you intend to do a randomized study with intervention and control, oftentimes during the feasibility stage, you don't necessarily need to randomize the patients. You just test the intervention, see if it works, okay? You're not comparing efficacy at that stage. You're just testing feasibility. But by the time you get to a pilot trial, it's really like the mini version of the final study. And if you intend to randomization, it's good to have randomization, but always make sure that you also include feasibility assessment. Here, you want to know, if I really want to do this study, how many patients I can get, how many patients will stick with the intervention, will they agree to randomization? If once you start saying that, well, some of you are going to get placebo, the patients disappear, then you know that this is not going to work. So recruitment and retention is very important. Trials fail oftentimes because we fail to meet the enrollment target. And therefore, you cannot draw any conclusions. Or you make the study too complicated so that patients disappear. Patients refuse to complete the study. You get lost, drop out, and therefore, you would not be able to get the study. So when you're planning these feasibility trials and pilot trials, it's in some ways a way by which you can learn from your mistakes and make improvement in the study design. So data collection. Well, I've mentioned that ideally, we're all very ambitious people. We want to collect everything under the sun. But is it possible? Because it may take so much time that for each study visit, the patients have to be with you for 4 hours. Well, after 1 hour, they get exhausted, they leave, okay? You want them to do a lot of tests, they don't do it, okay? Or even your coordinators get so exhausted. So you want to know what study procedures are acceptable and what would be the time requirement, what proportion of patients would actually stick with the study, complete those testing, how long it takes. Because if you realize that, well, I thought it's only going to take 1 hour, but it turns out to take 2.5 hours, then you can actually figure out what are the additional tests that you want to eliminate from the final study design. The other alternative is the most important tests are being done early. So it's okay if the patient missed the 6th test, but the 5 important ones are done. So you might arrange the sequence of those testing to improve your ability to collect essential data. Well, as I've mentioned, the whole purpose of the feasibility study is to help you do quality improvements so that the final study design is going to be better, more feasible, more acceptable to the patients, less burdensome, okay? And oftentimes, the initial feasibility study might go through several iterations. You might start with 5 or 10 patients just to give it a sense of how things are going to work out. Then you make some changes, you test it on another 5 or 10 patients until you get to a point where you feel quite comfortable, and now you start designing a proper pilot study. So for the pilot study, you're not talking about hundreds of patients. I've reviewed some study protocols submitted to me. It's like, this is a pilot study, but I'm going to have 500 patients study for 1 year. No, this is no longer pilot studies. This is a huge study that's going to cost you a lot of time and money. No, a pilot study oftentimes is much shorter in duration. So if the intention of the treatment is 1 year, you might in a pilot study only test 3 months or 6 months, but not necessary for the whole year. Now, the pilot study, because this is to inform the future study, you also want to get a rough estimate of how effective the intervention is. Because when you design a proper trial, you need to do sample size. So sample size estimation is based on your knowledge, your educated guess of how effective the intervention is compared to controls. But how do you know if this is something new? I've never done this intervention before. So the pilot study of 30 patients comparing to 30 patients might give you a sense whether the difference is 50% versus 10% response, or whether this is 30% versus 10% response, because that helps you to design your proper final study. So let's go through an example. Well, NAFLD, everyone wants to study NAFLD because no more Hep C, okay? So you want to see if I give every patient a Fitbit, would I actually motivate them to move more? And therefore, they would lose weight and the NAFLD would get better, okay? Now, if your ultimate goal is to make the liver disease get better, it's going to require a longer duration and a larger patient population. But early on, what you want to really know is, well, would the patients even wear the Fitbit, or would they actually take it and give it to their friends? Okay, that doesn't work. And how long is it going to take before patients are going to change their behavior and I start to see an improvement? And that is going to be important. So you're going to really do a test first. So you're going to do a feasibility study. So first, you say, I'm going to just have 20 patients. They're all going to have NAFLD. I don't really care whether they have fibrosis or cirrhosis or not. What I really want to know is, if I give people a Fitbit and I ask them to wear it every day for 4 days and upload the data, well, some of these Fitbits will automatically feed the data to you, so the patients don't need to do anything, but they still need to wear it, okay? I'm just going to see if my patients will wear it, or they wear it for 1 hour from 20 days, so I would be missing the data. Well, once you realize that, oh, the patients actually love it, and they wear it, and I get the data, then you can start testing for longer durations. For a few weeks, 4 weeks, they may wear it. Okay, now for 12 or 24 weeks, they may start losing the Fitbit, and that's a problem. So now you randomize the patients, and you make them wear it for longer. And in the intervention group, every week you send them a text message and say, good job, I see that you're moving. Okay, the control group, you don't tell them anything, okay? So it helps you, one, to see if people wear it for longer, and you might actually also include some efficacy measures. So you're going to record patients' body weight, not just the step count. You might actually get them some liver enzyme changes. Maybe you do a fibro scan before and after, so you begin to see whether you see a change in the steatosis score as well. So now let's move on to a bigger design. It's a magic pill. There's actually some Indonesian herbs that the village elder told you is very effective in making people lose weight. So you want to really design a proper clinical trial. Well, when you're trying to design a proper clinical trial, these are the elements that you have to think about. What's the background? Well, the background oftentimes describes the disease, the current treatment, and the pathophysiology. You have to have hypothesis, you have to have aims, the study population, specify your inclusion-exclusion criteria, the trial design, what's the intervention, what's the control, how long does it go on for, how you're going to randomize people, what are the endpoints, what, when, and how, what data you're going to collect, how you're going to analyze it, your sample size, and you put in your references. So let's start from the beginning. You have to have a hypothesis. So if someone comes to you and says, this magic pill would help NAFLD, you have to say, what's in this magic pill? Why Do you believe? What's the rationale? What's the laboratory data? What's the pathophysiological mechanism that would make you feel that there is a chance that it would actually alter the pathophysiology of NAFL? There has to be a basis for it. Then you have to say, what's the purpose of this trial? The purpose of this trial is to make NAFL be better. That's very vague. You have to define the primary endpoint. When you define the primary endpoint, is what, when, and how. And I'll go into that. You have your primary endpoint, you have your secondary endpoint, and you may have some exploratory endpoints as well. Now, endpoints are important because it influences which patients you're going to enroll into the study. Because if the purpose is to reduce fibrosis, and you only enroll patients with F0 to F1, how are you going to see an improvement? Okay? If you want to enroll patients and see if it would improve steatosis, the patients must have steatosis to begin with. Okay? So your endpoint can influence study the selection of patients. So now you get into the study population. You're studying NAFL, so obviously everyone has to have NAFL, okay? But you still have to specify inclusion criteria, exclusion criteria. Inclusion criteria, whatever you list, every patient must have to have all of them, not just half of them. But exclusion criteria is different. You meet one exclusion criteria, you're out. I'm sorry. Sometimes we want to be perfect. We want to have a very clean, homogeneous population. But if you try to be very strict, you find that you can't enroll any patient because majority of your patients don't meet all of those criteria. So you got to think through it. And also, if you make it too stringent, your results will not be generalizable to the patient population. So oftentimes the things that we think about is demographics, sex, age, race. We also think about how do we actually confirm the diagnosis of NAFL. Does everyone need to have a liver biopsy, or is it based on non-invasive assessment? What about the stage of the disease? What about comorbidities? What comorbidities would we allow? And of course, every patient has to agree to consent. And in most clinical trials, we will exclude patients who are pregnant because, in particular, if you have intervention for which we don't really understand the safety in pregnancy. So for a NAFL study, you will specify how you make the diagnosis of NAFL. Let's assume that you require biopsies, okay? Well, what if the patient had a biopsy? So when does the biopsy need to be done? Does the patient have to have a biopsy now, just before entering the study, within 1 month of entering the study? Well, ideally that's good because things could change. But if the patient had a biopsy 4 months ago, the patient's not going to agree to another biopsy now to get into the study. So you say, for practical reasons, I'm going to accept a biopsy up to 1 year before enrollment. But what if the patient lost 10 pounds after that biopsy? So now that historical biopsy may not be reflective of the current status. So you have to think of the test, the time window that you allow that test to qualify the patient to be in the study, and how severe the NAFL score, the NASH score, as well as the fibrosis score, because it depends on what is your primary outcome. And are you intending to have central review of all the biopsies? If that's the case, you need to be sure that if a historical biopsy is going to be used for qualifying a patient, am I going to be able to get access to those slides? If I can't get access to those slides, the central pathologist cannot read it, and that patient ultimately cannot be counted. So these are practical things that you need to think about. Well, what about exclusion criteria? Generally speaking, you have to exclude patients with other liver disease. Well, Hep B and Hep C are easy, but alcohol is difficult, okay? Well, you have to decide how much alcohol is acceptable, and you have to decide how you're going to collect that history. Because you asking the patient whether the patient had been drinking or not might give you a different answer from your coordinator asking the patient. Sometimes the patients want to be honest to you. Sometimes the patients are afraid that you will scold them, so they'll be honest to your coordinator but lie to you. So you have to figure out, and you have to really have a structured way of asking that question. Well, what about if the patient had prior treatment for NAFL or weight reduction? Would you accept those patients? What about comorbidities? Do you allow patients with diabetes, no diabetes, diabetes on certain drugs, diabetes not on certain drugs? Think about those things. Do you allow patients with decompensated cirrhosis? Maybe not initially. What about labs? So usually you define, okay, the platelet count should not be less than 60,000, for example. Hemoglobin must be more than 12. What about the creatinine? Think about all these reasons. And of course, we'll never enroll patients with serious comorbidity that might actually kill them before the study finish. So why do we need to care about all these things? Because ultimately, you want your study to be published. And when you write up the paper, you have to fill out this consort diagram, which starts by how many patients meet the criteria and how many patients are excluded from the study because of whatever reasons, and how many patients are randomized to each group, how many patients after randomization actually get started, because some patients after randomization to placebo disappear. You can never find them again, okay? So you need to specify them. How many patients finish all the study visits, as well as the post-treatment follow-up, and you have to include those, and you have to really explain why on the way you start with 100 patients and you end up only with 60. What happened to those patients? Are you hiding those patients and the results, or for what reasons? So then you have to think about the study design. Because it's a randomized controlled trial, you have an intervention. So now you have a magic pill. It's easy. So you have a magic pill as the intervention, and you got someone to make a identical looking placebo pill, and that's your control group. But you have to then sort of say, well, what's the dose? What's the duration? And under what circumstances would we actually adjust the dose? Because maybe because of side effects, you have to reduce the dose by half. You have to describe all these things, okay? How are you going to randomize? Nowadays, we all use computer-generated software to randomize the patients. And you also have to think, am I going to stratify the patients? Well, what do I mean by stratification? Because if you're doing a very big study, let's say, again, you're studying NAFO, and there's something about the pharmacological action of this drug that may make diabetes patients respond better, okay? In which case, you want to make sure that the number of patients with diabetes in each group, the intervention and the control group, will be balanced. So you might stratify that all diabetic patients are separately randomized from all the non-diabetic patients. So that in the end, I would have balanced proportion of diabetic patients in each of those arms. But if you make it too complicated and you want to stratify by diabetes, you want to stratify by creatinine, you want to stratify by history of cardiovascular disease, then the study becomes impossible to enroll because you have too many strata, and you find that you cannot have enough patients. All right. You also have to, during the course of the study, collect data on adherence. No matter how good the intervention is, if your patient is not taking the pill, it doesn't work. So think about how you're going to do that. Oftentimes, it is based on asking the patients, but do you trust the patients? Or if you don't give the patient a diary and you see them a month later, they're guessing, well, I am a good patient. I don't think I miss any days. No, in general, we give the patient a diary and ask them to record every day whether they are actually taking it. There are more sophisticated ways, such as electronic caps, where there's a computer chip to collect those information, but those are very, very expensive. You also have to, when we talk about adherence, it's not just to the intervention, but also adherence to the study protocol, which means that they don't have missed visits. They do all the tests that you ask them to do. Questionnaires. That's one thing that if you don't collect the questionnaire in your hand, the patients don't fill them out. So you need to make sure about that. Now, what about concomitant medications? What are allowed and what are not? You have to collect all those information because they can influence the response. They can also influence side effects. And if you expect that the treatment might have some side effects, or that you're treating a serious illness, some patients, the disease continues to deteriorate, you also have to think about, do I need to have rescue therapy for managing side effects or for disease progression as well? Well, randomization is very important. And the purpose of randomization is to make sure that both groups are balanced. And it has not you need to make sure that's totally random. So most of the time we rely on the randomization softwares, computer-generated software, so that you cannot guess. Because if you know it's A, B, A, B, A, B, you would unconsciously be biasing. I want this patient to get treatment, so I'm going to really get another patient to come in first and get the control so that this patient can get treatment. So you don't want to actually be able to influence that. Now, you all know that when you analyze the results, there's the intention to treat analysis, the ITT, and the protocol analysis. So when you randomize the patients is important, because if you randomize too early, as soon as you get the patients to come in and you randomize, and so this patient is going to get treatment, but actually the patient disappeared. The patient didn't come in to start treatment. That patient has to be counted as the treatment group because you had already randomized. You can't give that number to another patient. So you want to be careful that you don't do it too early. And I talk about the randomization. So for example, if we want to see if the treatment reduced fibrosis, so you want to make sure that the proportion of patients in both arms are going to be balanced in terms of having advanced fibrosis. Because if you have more F3 and F4 in one arm than the other arm, it would completely mess up the results. Primary efficacy endpoint. I've talked about what, when, and how. I've seen fellows say, my primary outcome is to improve NAFO disease. What is meant by improve NAFO disease? Make them live longer? Prevent them from requiring a liver transplant? Prevent them from getting liver cancer? Well, then you need 20 years of follow-up. Or am I improving symptoms? But majority of those patients don't have symptoms. So you say, okay, I'm going to pick histology. Well, that sounds easy, right? But no. What I mean by improving histology? You want to reduce steatosis, or decrease the NAS score, or decrease fibrosis. Well, if you say decrease fibrosis, well, decrease fibrosis by one stage of fibrosis or by 2 stages, okay? And you have to think of all these things. What if the fibrosis is less, but the steatosis increase or the NAS score increase? Is that still going to be considered a response? So you have to define all that, and you have to make sure that you determine when that biopsy, the second biopsy, is going to be done. Whenever you use histology, make sure that there is another complicating factor. Well, if your patient agreed to be in a study and then refused to get a second biopsy, then you cannot assess the primary outcome. So that's the tricky part of using histology. And you also need to have adequate size biopsy and slides that your pathologists say will be quality for read before you can use that to assess your primary outcome. So you have designed the intervention and all that. Now you have to figure out the study visits and what data you're going to collect. And this is, you want to see the patients often enough so that you can collect the data, but also you don't want it to be overly burdensome. So usually the study visit schedule will include clinical assessment, labs, maybe questionnaires, and also assessment for side effects. And you also need to record the dispensing of the drugs. And typically, if you have patient-reported outcomes, you have that as well. So this keeps moving back and forth. Okay, so this is usually the study visit schedule where you list the timing and whatever test. And you know that in real life, patients don't always can come back on exactly the day, or the next visit might happen to fall on a holiday. So you also have to define the window of the visit, plus or minus 1 day or plus or minus 3 days, so that you won't be considered to have protocol violation. Well, the last part is statistical considerations. Well, that's what we think would be the last part. But in fact, that should be very early on in planning a study, because if you don't think about all these things until you're done with the study, then everything is going to be messed up. So you have to have sample size calculation, and for each aim, you have to figure out how you're going to assess what statistical method you're going to use for analyzing. So you have to have sample size estimation, what population you're going to use for analysis, intention to treat population, per protocol analysis population. You have to have a plan. You're going to have missing data. How are you going to deal with missing data? And are you going to do subgroup analysis? Are you going to do interim analysis? And that will all impact the statistical analysis plan as well as the sample size that you require. So sample size is very important because that help you determine whether you actually can have adequate power. And that's predicated on your estimation of how effective the intervention is compared to the control. So if they're very, very different, 50% versus 10%, you don't need a big study. But what if it's 30% versus 20%? You need a huge study. So oftentimes we start by being really honest and we say, well, I don't think it's really going to be magical. So maybe 30% versus 10%. And then you do the sample size estimation, you realize, oh my God, I'm going to need 150 patients. Well, but I actually don't have enough money to do a 150 patient study. So what do? Well, now I'm going to fudge. Instead of I'm saying 30% versus 10%, I'm going to make the magic pill be more magical and be 50% response compared to 10%. Or now, I only need 40 patients. So that's easy. I can do a 40 patient study. But the magical pill turns out to be not so magical. And now you wasted your time and the study is not useful. So think about all these things. Patient dropout. So am I going to lose 5% of patients or 10% of patients so that you account for that and you add it to the sample size estimate as to how many patients you need to enroll. You also have to think about secondary endpoint because if you intend to do subgroup analysis, you have to determine whether your ultimate sample size would allow you to do subgroup analysis. Well, Jess has also mentioned garbage in, garbage out. So if you don't think about all these things carefully, then you're going to waste your time in going through all the exercise but not getting the results that you need. So it's extremely important and engage the statistician upfront early because you'd want to make sure that your effort is not going to be wasted. So thank you, and